Okay, welcome. And I've came up, when asked what I will talk about, I came up with a grand title. It's much easier to come up with a grand title than to actually assemble all the information. I'm afraid that it may, towards the end, not fully come up to the management relevance, especially when I won't talk much about the indexing system that's being used in the Baltic for characterizing the quality, the environmental quality of the benthos as part of the management according to the Water Framework Directive. But I think that you've heard some of that from Caroline. And I may say a few words about it at the end. But let's start with a background about the Baltic, which I think is important. Sweden has the longest coastline, the largest area of territorial waters in here, and the largest economic zone of all the Baltic states. So we have a considerable stake in managing the Baltic. And if you look at the Baltic, at the water depths, the bathymetry, you can see that the Baltic, as seas goes, is shallow. The mean depth is less than 60 meters. So, and especially the entrance area to the North Sea through the Kattegat is quite shallow. And this has important implications for things like the salinity of the Baltic. And you see the greatest depth are here about 250, here a little over 400, about 300 here, but average less than 60 meters. And the various subdivisions I'll talk about, the Baltic proper. The Gulf of Bothnia divides into the Bothnian Sea and the Bothnian Bay. And then you have the Gulf of Finland and the Gulf of Riga. And there is one important thing here. And, okay. Hey. Welcome. And that and that's you could see on the previous picture. No, sorry, the previous one. That there are sills between most of the basins, but the Gulf of Finland is directly connected with the Baltic, deep areas of the Baltic proper. So it doesn't have a sill, and that is an important aspect of that. The inlet areas here are very important. Most of the water exchange is here through the Great Belt. But when you get really salty water in, it often comes partly to a large extent through the Sound, which is somewhat deeper except for the sill. And this situation where you have a Baltic with a lot of precipitation over the sea and the drainage basin, so you have a lot of fresh water coming out. Then you have a countercurrent of salt water coming in. And this results in a situation where if you go along this line, you see that the surface salinity is about 2 up here, and then increases to 5, to 6, 7, and down here, as soon as you get into the Sounds, you get above 10, and then along the Swedish west coast, 15, 20, 25, and even 30 here. So, but even 30, as you know, isn't quite fully marine salinity. But you also see that along the line here, if you go deeper, you have higher salinity. And that's because of this inflow of salt water, which is heavier and will therefore end up in the deep areas. And this inflow can take two forms. Sometimes there is an outflow on the surface and an inflow at depth in the sounds. But the really great exchanges happens when you have a period of inflow all through the sounds. And that is if you've had a period of high pressure over the Baltic. So you have a low water level. And then you get a low pressure coming in with sucking in water from the North Sea into the Kattegat. And then preferably some kind of storm pressing the water in. Then you can get a great inflow of quite high salinity. The largest recorded one was 300 cubic kilometers in 1951. And then when that happens, you get an inflow all through at least the Gothland Deep here, and quite higher salinity than you had before, and also higher oxygen content. And those intermittent occurrences are very important. I won't talk much about that, but I'll just note that they used to happen every 2-3 years back in the 60s and 70s. But from the 80s on, they have become quite rare. They're now 10-15 years between them. And when you talk to the meteorologists and physical oceanographers, they don't know why this change has happened. If you ask them, could it be connected to climate change? They will say, yes, but we can't prove that. So, if you look at a particular point, for example here in the Baltic, you will find that you normally have a situation in the winter where you have low temperature all the way down to the halocline, where you get the increase in salinity. This is salinity. But in summer, you get a thermocline at 15 to 25 meters about, where above which temperature is much higher. But below here, you have an area, a water layer between about 60 meters up to anything between 30 and 20 meters, which is cold year-round. So some of you may felt the mud that we got out of the Baltic, and even in the middle of the summer, it can be extremely cold, 2-3 degrees centigrade only. And I apologize for the Swedish here. There are 1 or 2 slides that are Swedish. An important point here is that if you look at salinity and the freezing point, you will find that the freezing point is 0, of course, for normal for fresh water, but drops to something like minus 1.8 or so for fully marine water. But the temperature at which density is maximal is about, as you also know, 4 degrees for fresh water. But that drops as salinity increases, and somewhere around 25 or so, you'll find that density is maximal just at the freezing point. So this means that in the Baltic, most of the Baltic proper, you have maximum density at around 2.75 or so. Again, explaining why you get only 2-3 degrees centigrade in the in that intermediate water layer. The even deeper water in the Baltic is often a little warmer, but it's so much salter that it is still heavier and therefore lies below. And I've talked so much about Baltic salinity because it is an important stress factor for the organisms. Most aquatic organisms have an evolutionary background either from freshwater or from fully marine waters. And therefore the intermediate salinity of the Baltic is a stress factor for most of them. One common example is stunted growth. You'll find here, this is from Remane 1934, who pointed out that Macoma baltica becomes bigger at higher salinity, and this is even more obvious for Mytilus, that it grows to a larger size in full salinity. And this is not, it's very common for Baltic organisms. And you have a similar thing for some of the freshwater organisms, that they're actually somewhat smaller when they penetrate a bit into the Baltic. Another feature which partly can explain why they don't grow so big is that they tend to have a higher energy expenditure. And there you find two explanations for this in the literature. One is that they have to use so much energy for osmoregulation, that explains it. But I've also seen the argument that no, that doesn't explain it. You can calculate how much energy is used for osmoregulation, and that's not enough to explain the increased energy expenditure. So then the argument would rather be that they're living in a suboptimal environment, which means that all kinds of cellular mechanisms have to work at greater speed to keep the physiology okay. And also the stress from salinity means that there is very often lower tolerance to added stress. That used to be a major type of experiments that were done, for example, out at Aske, that you would have organisms in different salinities and test how much can they take of heat stress or cold stress, how much can they take of low salinity or so. And very, very often the result was when they're not in their optimal salinity, they can take less additional stress. And this also goes for a lot of pollutants, of course. So let's talk then about the components of biodiversity. Biodiversity, as you know, is an expression that has many components, and the one you most often talk about is species diversity, the number of species present in the community. But habitat diversity is also an important component. Habitat diversity, of course, means more or less automatically more species, because with many habitats there are many, there will be more species. And then also genetic diversity, and this has both a within species and within species and population aspect, but also between species, meaning that biodiversity is greater if you have organisms with very different genetics, organisms from different phyla, for example, compared to when you have the same number of species but from the same phylum. But genetic diversity within species and populations, of course, is very important, because if you have low diversity, genetic diversity, it means simply that you are less able to adapt to changes in the conditions. There is less for variation for natural selection to work with. And there are studies from the Baltic showing that generally marine species that penetrate into the Baltic tend to have lower genetic diversity in the Baltic than outside. And very often the greatest change in genetic diversity occurs in the area of the Sounds, where you also have that considerable drop in salinity. Food chains in the Baltic look strange to most people because you have this mix of organisms that are of clearly marine origin and of freshwater origin. You have perch and pike swimming and searching for their food in bladderwrack belts, and perch will eat gammarus, which is of marine origin, and so on. You have the planktonic phase is generally dominated by marine organisms, but there are some of freshwater, for example, some cladocerans. And the benthos has a mixture, and I will return to that, of marine and freshwater organisms. And the marine organisms, here you have, and we'll return to that a little bit, here you have the number of invertebrates in the Baltic going from the Skagerrak into the inner parts of the Baltic. The exact figures here are quite old. They go back to the 50s or so, and more species have been found since then. So if you made a really up-to-date list, it would actually have more species than this in all areas, but the progression would be the same. And you see many typical Baltic organisms. Macoma occurs, the Baltic tellin, as it's called, occurs, Macoma baltica occurs, except in the very northernmost end. Balanus improvisus, which is actually an introduced species, occurs also almost all the way in. How far north you can find cod varies with the cod stock in the south, but this is more or less the maximum. Fucus also occurs about to this, but not the northernmost ones we now know may be another species. I'll return to that very, very briefly. Mytilus, again, not necessarily Mytilus edulis, but it's some sort of genetic mixture between Mytilus edulis and Mytilus trocylus that we have in the Baltic. You see there are some species that are more or less excluded from the Baltic, like sea urchin and sea stars occur to the island of Bornholm, not any further. Crabs are used to occur only to here. Now we have an abundant population of an introduced American species spreading here, and it will undoubtedly sooner or later turn up at Askja as well. The situation in the Baltic, where you have a salinity here and you have freshwater forms dropping off, very few of them taking more than 5 or 10 per mil, and then marine forms dropping off very quickly as soon as you go below about 30. But a few of them surviving, like we saw, up to down to 2 or 3. And then we have a number of species that are considered to be specific brackish water forms that are really adapted to brackish water. And when you enter a European estuary where a river reaches the sea, you'll find a similar situation with a minimum somewhere here around 8 or 10. And this is really, this curve here can be compacted like this, and it's then called Remanes curve, with the fresh waters going down, the brackish water forms, and then the marine ones coming in. And this one has been extended to really high salinities as well. And when you compare the number of species in the Baltic and the more marine waters outside, you see that even for things like planktonic algae, you do have fewer species in the Baltic. But for macroalgae, it's more dramatic. For fish, it's also clear. But the most dramatic decrease is the one we saw already for macro invertebrates. If you were to add microscopic invertebrates, there would be many more. And in fact, when you go to the eukaryotic unicellular organisms, there is a profusion of organisms that are, in many cases, are only known from their DNA. We don't really know what they look like. So there is an enormous diversity that we are only beginning to understand now. And the same goes for microorganisms. But generally, it's among the bigger organisms that you see the greatest reductions in biodiversity in the Baltic. And when it comes to the inner Baltic and the sediments, I mentioned out at Aske that if you want to find a community where you have only 4 or 5 species, as you find in the Baltic, in the world ocean, then you have to go to a period such as this, the end Permian extinction, which was the greatest extinction event known in the history of life on the planet. Then you saw abundant high diversity communities such as this one, with maybe 50 to 100 species in them, reduced to 4 or 5 species. And this is similar in terms of biodiversity to what we have in the Baltic. So clearly salinity is a stress factor. Another important thing for the low diversity in the Baltic, of course, is the geological history. We would expect that if the Baltic could stay the same for a very extended period, a number of species would be created within the Baltic, would evolve within the Baltic, and we would gradually have a large number of species. This has happened, for example, in many inland lakes that are isolated for a long period. They become very species rich, things like Baikal and so on. But the thing with the Baltic is it doesn't stay the same for very long, and this is the history. The Baltic started out after the last glaciation as the Baltic ice lake. Then for a very short period, we had the Yoldia Sea with higher salinity. And what is now called Portlandia Arctica as a lead fossil. It's now only found up here. And then it became a lake again, the Ancelus Lake. And about 8,000, 7,500 years or so, the sea surface was increasing because of the melting of glaciers. So you got the sea coming in again here into the Littorina Sea. But then this had higher salinity than maybe twice as high salinity as we have today. But then the land tends to rise, and you get less water exchange, and now we have the typical Baltic situation since about 2,000 years. This stage of the Baltic has at one point been called the Nya Sea, which is, now that we know that Mya is Mya arenaria is introduced from North America, is maybe not such a good name for it. Where do Baltic animals and plants come from then? And I'll mainly mention the ones that are in the benthos, but I have some others on my slides. So you see things like Hediste diversicolor we saw out at Aske, Hydrobia ulve, and Macoma baltica. They are estuarine, everyhaline marine organisms who are well common in northwest Europe. But we should note that Macoma baltica, we have a somewhat different race in the Baltic, which is more related to some of the Arctic strains. And then we have typical European freshwater species. Oh, sorry. We didn't see many of those out there, but these you find, for example, you probably saw these, but not in the benthos that we looked at. And things like perch and pike. Then we have glacial relicts, organisms that occurred in these ice-dammed lakes, Arctic freshwater organisms like the large isopod Saduria, Monoporeia that you saw out there. And then you have some that are considered glacial relicts of a marine origin that occur today in the Arctic seas, but are found very seldom, say, in the North Sea. And here's one that was discovered just a couple of years ago, I would think is one of these glacial relics. This was misidentified for a long time until Elena Gorokova became suspicious and had tested the DNA, and it turned out it was entirely different genus than had been thought before. Pontoporeia femorata you've seen. In the southern Baltic you have Macoma calcarea. Halicryptus, for example, is very occasionally it's been found on the Swedish west coast, and there is, I think, one single find from a Scottish fjord, but otherwise it's absent from the North Sea. And an important thing is that Europe in the last glacial maximum looked something like this. You had huge glacier, and many of the organisms, and you had, because you had a lower level of the sea because of the glaciers keep binding up the water, so you had an entirely different coastline. And many of the organisms that are typical for the Baltic today were then pressed south, because up here was really Arctic conditions. And we've had a progression. We've had some 23 or so ice ages. And that means every time this happens, the organisms had to first flee the ice age down south, and then return again, flee again, return again, and here is a stretch of line with very few estuaries, stretch of coast, where it would be very difficult for a brackish water organism to find any place to stay. And this may be a reason why some organisms that have occurred in Europe, like Mya arenaria, the sand clam, it became extinct in Europe about 5 million years ago. And then suddenly it reappeared about 1200 or so AD, as about 800-900 years ago. And we can only suspect that somehow the Vikings brought it back from North America, where it had survived all the time. Then we have recent introductions and invaders. We have Mya is one of them. Balanus improvisus another, of Caspian origin rather than North American. Paludistrina or Potamopyrgus jenkinsi, a small gastropod from New Zealand. Rainbow trout. And then Marencelleria, where we have 3 species, all of them apparently North American, that have been coming into Europe in recent years, and which is now a very important element of Baltic fauna. The round goby, Caspian origin. The same goes for Ponto-Caspian origin, rather, Black Sea, Caspian Sea. And then you have Sarcophagus also from that area. And there are a number of more species that could be listed here, and there are some that are on the way that we expect to see anyway now, because they can survive we know they can live in brackish water. They've been found quite near the Baltic in fresh water. And then we have a single organisms that we really know has evolved in the Baltic, and I'm sure Lena's told you about Fucus radicans, which has evolved here probably about 1,000 years ago, and is now a distinct species, reproductively isolated from Fucus vesiculosus, its mother species, so to speak. But the Baltic has also lost species, so it's not only gained species. The gray whale, most of you know of it from Alaska, but it was scientifically described from a subfossil find from Uppland, not far north of here, in the 1850s or 60s. And then only gradually did people realize that it is actually the same species as is known from the Pacific. And but it is known that it has occurred along the Baltic coast, and it is thought that it may have been exterminated already about 800 or 1,000 years by the Basques, who were the first whalers. And probably due to the reduction of the polar ice, there was actually a gray whale recorded in Europe, I think 2 years ago. A single one suddenly appeared off the coast of Israel. Total mystery. But the only explanation one can think of is that it penetrated further into the Arctic than normally because of the withdrawal of the ice, and then it came out the wrong way into the Atlantic rather than into the Pacific. And then of course it would swim along the coast, along the western coast, eastern coast, searching for Baja California, where they breed, and end up in the Med outside Israel instead. We have the great auk. I think it was last seen in 1844 on Iceland. It's a big bird, which is actually, it was called a penguin in French, and this is why penguins are called penguins. But this occurred in historical times in the Baltic. The harp seal, in Swedish Grönlandssäl, was also found in the Baltic for several thousand years. And we know that it was a resident population because over time it got smaller, considerably smaller than the Arctic population. So it was clear that it was an indigenous population. And it actually survived the warmest history part of the Baltic history. So it wasn't climate that killed it off, but humans became more and more common, and it has been very commonly found in archaeological finds. So it's reasonable to assume that humans exterminated it. And then we have the sturgeon. And whether it's the European sturgeon or the Atlantic sturgeon, we should say has been exterminated, is a bit unclear, because if you look in archaeological finds again, before about 1500 years ago, you actually find the European sturgeon. And then suddenly, about 1500 years or so, you start to find the Atlantic sturgeon, which is otherwise known from North America. And no one can really understand it, but they've tested this with DNA. So it's pretty clear that that switch happened somewhere. And it was clearly before Columbus. But I don't think the Vikings could have brought over a 3-meter fish. I think it would have had to swim itself. And then, of course, the Atlantic sturgeon is live and well in North America. It's a bit threatened, but there are large numbers of it. The European sturgeon last bred in outside captivity in Dordogne in 1987. So that is really a threatened species. And in any case, as far as I know, the last time one was caught in the Baltic of either species was 1996. And then when you talk about biodiversity, I think it's important to have this perspective also. If you look at the enormous diversity of organisms, then you find that all fungi, animals and plants are really more or less the same in a genetic sense. We are very similar to plants or so if you compare to the enormous genetic diversity that you find among the prokaryotes, the archaea and the bacteria. And it's here you find the big things where you have this much lower diversity. But even among these organisms, when you start looking at bacteria, for example, you will find that, which we didn't know only 15, 20 years ago, that you have a different set of bacteria in fresh water and in marine waters, and the Baltic ones are intermediate in their composition. So you have a similar situation for them, but it is still not very well known. But we'll soon know. They're working at it in our department, Birgitta Bergman's group. They're looking at prokaryote genetic metagenomes in the Baltic. I'll skip that one. And we've spent some time studying the ecosystem effects of biodiversity. And why should one study the effects of biodiversity in a place where there is so little diversity? And my explanation, my motivation for that is that one needs to study effects over the full range of a factor. One needs to know the effects of biodiversity, whether there's little of it and whether there's a lot of it, to really understand it. And it's always easier to understand and study simple systems like the Baltic. So I think therefore it's really very useful. And one can study here the real system. Very often when you see people doing biodiversity studies, they've selected 40 or 50 species and put them together out of a species pool of maybe hundreds of species that could have been selected. Whereas in the Baltic, we have very few species. We can really study the real community, at least when it comes to things like the macroinvertebrates. And also, studies here have management relevance because benthos are used as an indicator in the Water Framework Directive. So it's one of those things that EU law says we have to study in order to judge the environmental quality of both fresh waters and the seas and the coastal seas. And this, of course, could be done equally well for plankton and phytobenthos. If you look at benthic organisms, you tend to categorize them in different ways. You can categorize them by size as microbiota. That is normally in the Baltic, we say that's anything smaller than about 40 microns. And that means you have protozoa, fungi, bacteria, archaea, and the occasional metazoans, mostly then small juvenile nematodes. You have meiofauna, which are in the Baltic, we generally larger than they're caught on a 40 micron sieve, but they pass through a 1 millimeter sieve. And this is a little bit different. In many areas, they use a 500 micron sieve to delineate what's meiofauna. And this then is generally small multicellular organisms, large ciliates, forams. Those are the groups that you'll find in numbers. And then you have macrofauna, which are things that are caught on that 1 millimeter sieve. And here you're almost only dealing with metazoa, but there are some deep sea protozoa that grow to several centimeters. So you can find also some and some forams that are so large that they can be found in that group. For example, the pyramids of Egypt are made of forams which grew to about this size. You don't find them there now. They're just fossils, but there are other protozoa that grow, the forams that grow quite large. And then you have megafauna, generally considered larger than about 10 centimeters. And this was a faunal category that was first introduced when people started to take photos of the deep sea. And those were things that were large enough to be seen on the photographs. You can also define them by the mode of living. Epifauna live on the sediment surface. endo or infauna lives buried in the sediment. And then you have a group called hyperbenthic fauna, which tend to swim just above the sediment surface. Of course, if a predator appears, they're normally quick enough to bury into the sediments to escape. But most of their activity is just above the sediment. And in shallow waters, you have quite a few organisms that are hyperbenthic at night, and then of course buried during the day. And you have a similar set of explanations for size for plankton, but there you tend to talk about sometimes nanoplankton, then you're dealing with small bacteria. Picoplankton, the very smallest photosynthetic organisms, many of them cyanobacteria. Nanoplankton, most of the unicellular eukaryotic algae. Microplankton, mesoplankton. And then you go to macroplankton, megaplankton. Megaplankton are things like big jellyfish and so on. And the nekton, the swimming fish and crustaceans. And as I mentioned out at Ask, originally when people started to look at the benthos in terms of microorganisms, microbiota, meiofauna, macrofauna, they envisaged them as separate groups. This has some relevance in sand, but doesn't seem to be true of softer sediments in the Baltic, like clay and so on, silt and clay. When you look at organisms, how they occur, you will find that things here like myarenaria, and you have Macoma baltica up here. You have Nereis, Hediste diversicolor, which you also saw. They occur, most of them, in the upper 5 centimeters of the sediment. Now in the Baltic we have Hediste sometimes goes down to 30 centimeters, and we now also have Barentseleria, which goes down deeper. But most of the macrobenthos in the Baltic is in the upper few centimeters, or like lying on the surface, like some Mytilus. Meiobenthos, on the other hand, things like these harpacticoids, ostracods, nematodes, but these are not to scale here. But you'll see that they are concentrated in the upper centimeter or so of the sediment when you have a muddy bottom. If you have sand, then they can go quite deeper, especially if it's an exposed sandy beach. Then, and it all depends on how deeply oxygen penetrates. The things that you do find below the redox layer in the anoxic sediment are mostly two groups of organisms. There are some species of nematodes, and there the general explanation, the belief is that they sometimes go up into the oxic zone to breathe, and then down to eat, and then up and breathe. But it's not really been well studied. And then you have forams. And it was discovered a few years ago that forams can actually denitrify, something that one thought only bacteria could do. And that probably explains why you can find quite a few forams deeper into the sediments and being alive. And then when you go to the real microfauna, it looks something like this, as illustration from Tom Fenchel, where you see some large ciliates, you see really small, really small bacteria, and you see some sulfur bacteria, you see some diatoms and so on. This is from a sandy sediment in quite shallow water. And when I became a PhD student, the task I was given, but maybe we'll take a break first before I become a PhD student. So I suggest we meet here in 7-8 minutes. Till 1970 när jag började som doktorand. Då fick jag alltså uppdraget att undersöka vad betyder mejofaunan i Östersjön. Sorry, I'll take you back to 1970 when I started as a PhD student, and my task was really to find out what is the importance of meiofauna in the Baltic. And I worked closely with a colleague, Sven Ankar, who looked at the macrofauna at the same time, and I'll show you some of. So what to do when you're a fresh PhD student? And meiofauna was, there were no publications. I think there was one publication actually from the Baltic, except from a few from Kilbite and in shallow sandy beaches more than anything else. So there were very few publications. And obviously, take some samples, learn the fauna, develop methods to sample quantitatively, and to estimate the biomass of individuals and communities, then design surveys to estimate aerial averages, and we started with the ASCII area, and then we tried to look at the Baltic Sea in some kind of an international cooperation. And also eventually we followed annual dynamics with relatively monthly sampling over time. And I'll give you a few examples of that. Methods, what I came up with was basically that using a grab was meaningless because you have this bow wave pushing things away, and a grab, as it digs into the bottom, also distorts the sediment, so you don't really have an undisturbed surface to sample from. What I came up with was a small corer like this, with which has a lid on top which is entirely open, so that the water flow through it is unrestricted. It turned out, when I actually tested this later, that it wasn't as good as I thought. So nowadays what I say is you have to use a rather larger corer, and even then it's not perfect. Even then it's better if you can have a diver go down and take the samples. But that is difficult once you get above about 30 meters. Because there are large restrictions on diving, and particularly the kind of diving where you need to stop on the way up to equilibrate your CO2 and the N2 in your blood. And anyway, learn the fauna, and the major players really are the nematodes, by far dominant in numbers, harpacticoids, and ostracods. Ostracods in the Baltic turned out to be often quite important in terms of biomass. And then there are some minor groups like this this turbellarian, and they can be important because they tend to be predators, so that even within this group of small organisms there are some predators. In 1971 we took a small boat and went around Asker and sampled with a grab and that corer you saw at a number of places which we had selected by randomly over the area. But we divided the area into 5 sub-areas, and within each of those we took random samples. And if you remember, when we were out there, here's the lab, and basically we sampled up here and around here. So this, it was about the way we covered with Aurelia compared to the total area. And if you start looking at meiofauna, what we found basically was that down to about 50 meters or so, we had a number of groups. And here we have the biomass, and you see that ostracods tend to be a very important part of it. Nematodes another, and harpacticoids also. Then you have smaller groups like Kinorynx, turbellarians, and temporary meiofauna. What is that? That is simply the small individuals of groups that later, when they are reproductively mature, are part of the macrofauna. But they're often, for example, Macoma, when it settles to the bottom, can be as small as 220 microns. So it's 0.22 millimeters. So it's clearly within the meiofauna. And it takes it, if you look at 30 to 40 meters, it takes it more than a year to grow to a size where it's actually caught on a 1 millimeter sieve. Whereas in shallow water, that goes in a few months. And then you see somewhere around 70, 80 meters, you start to run out, oxygen conditions are worse. Now they're virtually zero very often when you get down to 80 or so. But then we had somewhat better conditions. But still, when you get down to 120, 130, it's virtually only nematodes you find. And if you look at the annual dynamics of the pelagic zone in Asker, this is an old picture by Hagström and Larsson. You can see that the primary productivity in the plankton has 2 clear peaks. One, the spring bloom, dominated by diatoms initially, very often then by dinoflagellates. And then you have the summer bloom, cyanobacteria, but also lots of small pico cyanobacteria, not only the large ones that you see with your eye and that are nitrogen fixers. But when you look at the phytoplankton biomass, you see that you have a big peak during the spring bloom, but during summer you don't get much of a biomass peak. And a reason for this is that this spring bloom is grazed on by ciliates and by a few zooplankton, mostly rotifers. But most of that biomass is not grazed at all. It sinks to the bottom, where it becomes food for benthos. Whereas in the summer, you don't get a phytoplankton biomass peak, even though productivity is high, because you have lots of zooplankton that eat them. And when you have lots of larger zooplankton, mostly copepods, but also some cladocerans, then they will also tend to eat ciliates, not only phytoplankton. And why do we get a decrease later in zooplankton? Well, about this time, this is about August, late July into August and September. That's when you have the fish larvae in great numbers. So when the fish larvae come in, they tend to graze down the zooplankton, and you have that kind of succession. And sometimes, after they've grazed down the zooplankton, you can have a new peak, autumn bloom of phytoplankton, but that doesn't happen every year. How does this tally with what happens on the bottom? And I'll only do the meiofauna here. Here we have meiofauna samples taken from approximately, oh sorry, I should have had another one here, but I don't think I have that. Yes, I do. Okay, when you look at the meiofauna, this is sampling every month from October 1988 to October 1989. So altogether it's a little over a year, but the year then starts here and then runs around to here. And when you look at the most common ones, the nematodes, they decrease until April, and bang, down comes the spring bloom, and you get an increase in numbers. And that increase occurs particularly in the surface sediment, the 0 to 2 centimeter layers, but also to some extent in the 2 to 4. When you look at most of the other ones, you don't see very clear annual, but one group where you see it very clearly are the harpacticoids, small benthic copepods. And this is the total picture. You see a peak in winter and then a new peak in spring. But when you look at it more carefully and divide it into the two groups, you find that this is simply one group is a winter species, Pseudobradia, which is a small yellowish, very common one. But and then you have Microarthridion, which is a larger one, and that tends to reproduce starting in April when the spring bloom comes in. So you have two types here. One which has the same as most of the nematodes. You reproduce during the time of most abundant food. But you have one which is clearly a winter species and breeds during the winter. Yes?
What do they eat?
Is there abundance? Well, as far as we know, this is from near the buoy station B1, you know what that is, where the monitoring takes place. And that's deep enough that you don't have much in the way of benthic microalgae. But you do have the sedimentation from above of algae. And then you have detritus during the rest of the year. When you look at the macrobenthos, Hans Cedervall did a study of one particular station in the Aske area, which is about slightly over 40 meters depth. And he found a community which had essentially those 6 species dominated by Pontoporeia femorata and Monoporeia affinis. About 90% of the production over a year. Less if you look at biomass, but if you look at production, they stand for about 90%. At that station there were very few Macoma, as is common when you have lots of these amphipods, because they eat very small macrobas. So as long as you have many, you don't get successful settling of macroma. And then you have the predators in the system, Halicryptus, the scaleworm Bulgides, which I don't think you saw at all, but which used in those days when it was about 1 per mill saltier out there, it was quite common. And then Saduria, of course. And they all eat these others, but only Saduria is able to crush macroma. Bulgides, of course, a big Bulgides can eat a small macroma, but it'll eat it whole. And it's possible, it's not impossible that even Halicryptus could eat a really small macroma, but at a bottom like this, the main food will be the amphipods. And again, of these species, those two have larvae and spawn basically in the spring. And then they have larvae that are spread with the water. And I think macroma is really recruited from shallow areas. I don't think a lot of larvae are being produced at these deeper stations. But you have lots of macroma shallow, and some of them, of the larvae, will end up in the deep areas. Then you have Halicryptus, which is really unusual because it has larvae that are not planktonic. They look entirely different from the adult, but live on the bottom all the time. No one knows what they're eating down there, because it's not easy to be a predator if you're really small. And then you have the ones that produce young that look relatively similar to themselves, so they're not larvae. And that's the amphipods. They have a marsupium where the eggs are carried for several months, from something like November when they spawn, when they breed, until March, April, when they are released. So generally speaking, the young are released at about the time when the spring bloom ends up on the bottom. And then they can grow quite rapidly initially. And Hans Cederwall did an annual study of this, and he saw that they generally grew very rapidly until summer, and then growth more or less stopped. And then they would get another spurt next spring, and by the time they were 2 or 3 years old, they were ready to breed at this station. In shallower water, they can complete the life cycle in 1 year. And then Saduria, it is also it's an isopod, but belongs to the same big group of crustacea, the Paracarida, that has marsupia, where they carry the young. And they can also they carry the young for quite a long time, but as far as I know, you can find them carrying young more or less all through the year. But I haven't really checked that very carefully, because you would expect, for these, we know that the kind of breeding tied to, clearly tied to the annual cycle, that happens down to about 40 meters or so, where they can see some light during summer. Deeper than that, they can breed year-round because they simply don't know when it's spring or when it's autumn and time to breed. So, and I can imagine that it might be the same here, but I haven't checked that. And again, we had a discussion here. I suggested that these can breed again, whereas Caroline told me that no, they can't. When you actually look at some of the literature, it turns out that, at least the way I look at it, it isn't proven. There are some who have reported that they can actually breed and survive, presumably to breed again. But it seems that most of them die after breeding. So it may be that they can actually breed again, but like Swedish salmon, they probably functionally they breed once and then die, even though a very few may survive to breed again. And if you look at in the Asker area, and here we have different depth zones from 9 meters to 50 meters, and the biomass, you see that macrofauna biomass was mostly Mytilus in the shallow area, and the Macoma. And then Pontoporeia, the two species with Monoporeia and Pontoporeia. Monoporeia in the shallower area, Pontoporeia deeper. Those four are the dominants. And Mytilus, of course, occurs mostly on sandy sediments. It will sink into the mud and not do well there. And then you have a few others, as you've seen. And if you look at the distribution of macrofauna taxa versus death, and I told you about this at Asker, you'll see that in really shallow water you can find up to slightly over 20 species of macrofauna. That's insects and mollusks and crustaceans of various sorts. But as you go deeper, the number decreases. And down here, of course, you tend to get into anoxic zones, and you get to very near zero. And this is very different from the Swedish west coast, for example, or off Norway, where you have many more species. You would start out in shallow water at maybe 30 or 40 or so, and then you will go to more species as you go deeper. And the reason, general explanation why you have more species in marine areas as you go deeper is that the conditions are much less variable. There's less variation in salinity, less variation in temperature. And that makes it easier to survive for more species. Whereas here, it's rather the other way around, that in the deep areas, in the shallow areas, you have lots of species adopted to living in the phytobenthos. They don't survive deeper. And you also have a lot of freshwater organisms, and they don't do well at depth. And of course, that is an important difference. I'll skip that one. And when you look at both the macrofauna and the meiofauna, you can actually make schemes like this. How does it tie together? You have organic matter being imported from and microalgae being eaten by SAM. And then you have bacteria breaking down organic matter. And you have loads of protozoans, meiofauna, the various species of macrofauna that eat this. And then the predators that eat the macrofauna, the main ones are here. And then, of course, you have fish. And you also have some birds. Most of the birds eat fish, but some of them also eat mollusks, for example. And even some of the crustaceans and so on. And so you see, even an extremely simple ecosystem, as I've been telling you, can become quite complicated when you start taking all the interactions into account. And if you're going to look at all the different species of bacteria and so on, it just mushrooms. It's the diversity, even in this, or the number of trophic relations is enormous. I told you about temporary meiofauna, and that may seem as an oddity, but if you look at a total number of macrofauna and then divide it into those that are found in the meiofauna fraction and those that are found in the macrofauna fraction, you've come up normally with something like this. This was based on that study in the Asker area, some 36 or so samples. And you see that for many of the groups, oligochaetes here, Monoporeia, Chironomids, Hydrobia, Mytilus, Macoma also, virtually all of the number of individuals are in the meiofauna. And even for the total, the number found, you found an average of maybe 3,500 per square meter in the macrofauna, but it would be about 45,000 in the meiofauna. In terms of biomass, of course, it's the other way around. All these small ones look insignificant then. But the small ones, of course, grow more rapidly, have a higher turnover, higher respiration rate per biomass. So they're more important than they are in terms of activity than they are in terms of biomass. But if you want to understand the population dynamics of some of these, you clearly have to take into account also the meiofauna fraction. Then we turned to and tried to get an international cooperation to sample the whole Baltic. It didn't quite turn out right. We didn't get results from the southern Baltic and the eastern Baltic. The Russians were happy to take our corers, but they never reported any results of the Soviet people. And I don't think this had anything to do with the scientists. I think they were just forbidden to send anything to us. And it turned out actually at one point when I talked to Swedish military that they were very keen to have our results. So there was maybe there was some reason for the Soviets not to send it over, because apparently it has some importance what kind bottom type you have when you're looking for U-boats and so on. So, but these were those old days. But anyway, we collected ourselves samples from a considerable area of the Baltic. And when you look at those areas, this is the macrofauna diversity going from here around the south of Gotland up to the Bothnian Bay in the north. Then you can see that in the Baltic proper, you had fairly high biomass, and this is what we had in the Asker area, the Asker survey. But most of the samples in the Baltic proper were deep ones, where we had very little because of oxygen deficiency. So these are down to 80 to 120, 150, 180 meters. And then when you go here's the area of the Stockholm archipelago or so, you go into the Bothnian Sea, you have initially rather high values, but then Mytilus disappears, and then somewhere here you start finding very few of anything. And you see that around here you have a species minimum, and you actually get occasional samples in the Bothnian Bay where you have oxygen, there is no toxicants around or anything, yet you don't find a single macrofauna individual in a whole grab. Very special. But when you do find, they're either monoporeia or saduria. Or rather, I should say, you have either monoporeia or you have monoporeia and saduria. And this means, of course, that I showed you the Riemann curve before, where the salinity minimum of species was around 6 or 7 or something like that. But when you look at the benthos, the deeper benthos here, the minimum is actually up in the Bothnian Bay, where you have 1 or 2 species. If you look at biomass, and this, you have a similar figure of decreasing as you go north, but this is a logarithmic scale. So this goes from typically 100, but sometimes up to several kilos per square meter, down to here, where the maximum is about 5 grams per square meter, and many are below 1 gram per square meter. So it's a really dramatic decline in macrobenthos. But if you look at meiofauna biomass, you see that it goes from around 10 or so down to something like 3 or 4. But it's not at all as dramatic as for the macrobenthos. And again, you also see that at these deep stations, you tend to have a reasonable amount of macrobenthos, meiobenthos in some of them, even though you have very little macrobenthos, and those then are mostly nematodes. So this is contrasts the two. Very steep decrease in the macrofauna, much slower decrease in the meiofauna. And this is actually exactly this picture you find if you, rather than having a geographic gradient here, if you were to have a depth gradient going in the open ocean, going down from shallow waters to the deep sea, you'll find that macrobenthos decreases very drastically, but meiofauna not at all as drastically. So you find that when there is very little food, it turns out that it's better to be small, because what you eat are maybe individual bacteria or small colonies of bacteria, very small particles of detritus. And if you're small, it's easier to make a living that way. If you compare that with Baltic primary productivity, and this goes again from this is around Gotland, and then going up to the Bothnian Bay, you see there is a very drastic decline in that as well. But it's not it's less drastic because this is not a logarithmic scale. So it's not as drastic as the decrease in macrobenthos. But when you try to calculate approximately how much energy is used by the meiofauna and is used by the macrofauna, and then add them together, you'll find that the benthic activity decreases at about the same rate as the production of organic matter in the surface water above. Nowadays, of course, it's easier than it used to be to collect monitoring data from many nations and so on. Most of the data are free, and this is one paper from Gogina and Settler, where they've used collected data. This is just a preparatory slide. This shows the salinity, and this shows the sediment type based on available sediment charts. And you can see that hard clay, mud and clay, there are large areas of mud and clay, but they're not totally dominant. There's a lot which is hard clay, basically a glacial clay which is exposed, and sand, and you have some that are a mixture of many types, and you have bedrock. Of course, if you look at this map here, off Askja everything is bedrock, and we know that's not true. So it's impossible to have all the details in a map like this. And if you look instead, at the depth, you've seen this essentially in another form. You see the deep areas here, and again salinity with slightly higher salinity in the deep areas. Then they've used a huge amount of monitoring data to make maps of a few species, and the real data are these points and the rings, which indicate how much they've found. And then they've tried to make a model that will calculate what is there between the data points. Very modern. And this is the result for Monoporeia affinis, but they still called it Pontoporeia affinis, but Monoporeia affinis. And you see that it occurs down here, essentially to the Danish sounds, but it only becomes common further in to the Baltic, and then it occurs all the way up. And they've tried to use this data to compute what it will be between the data points. I'm a little bit hesitant about some of the deep areas here. I don't think it's likely you find anything, but their model says there is. This is Macoma baltica again, occurs from the sounds all the way up to about here. And Halicryptus, and here's where the modeling starts to break down, I think, because Halicryptus occasionally found on the Swedish west coast, but it's quite rare there, inside the sounds all the way up to about here. There, I think, is another find, but I'm not sure if that's a real one. But into this area, we have found it earlier, somewhat further north here as well. But then their model suggests that it should occur here in some, but I really don't think you can find it there because of the low salinity. And then you have more marine species like Arctica islandica, which you find on the Swedish west coast and in large numbers down here in the Kiel Bight and the Arkona basin, but not further in. I'll skip that. Well, based on the type of trophic schemes that you saw previously for macrobenthos and meiofauna, you can make energy flow models based on annual averages for areas in the Baltic. And I did this in 1984, where I looked at primary production. I used mesozooplankton, plankton, and invertebrate predators, essentially mysids, pelagic fish, and then bacteria, meiofauna, macrofauna, demersal fish, and tried to make a scheme for the Baltic, which came out with these figures are grams carbon per square meter per year. And I could make then a scheme for the Baltic, where you see that only a small part of this, a fairly large part actually, sediments out. Some is filtered out, but you still get rather low production in the benthos, higher production in the plankton. And of course, the highest groups are the bacteria. And when you compare the Baltic proper with the Bothnian Sea, you find that the Bothnian Sea is similar to the Baltic, but most of the figures are about 2/3 of the Baltic, except for the benthos, because here you have oxygen deficiency in the deep parts and no macrofauna. Here the deep parts have quite a lot of fauna. So the benthos becomes proportionally much more important there. But when you go up into the Bothnian Bay, an important thing happens. Virtually all the sediment, the deposit feeding bivalves drop out. You do have an occasional Bacoma there, but the numbers are so low that based on the averages for the whole area, they're essentially not important. And then you get a much lower importance of the benthos. And this is where also meiofauna is much more important than macrofauna in terms of the energy flow. And then I started to wonder, now the Baltic is a very simple ecosystem. What does that mean if you compare to the North Sea? And there was a model of the North Sea that had been published some years before by John Steele at the Aberdeen laboratory. And if you compared the two, in fact, you found very similar figures in many of the places here, with one difference. The major difference was that Steele didn't include the coastal areas, and most of the North Sea is so deep that he didn't have much of an importance of filter feeders. Otherwise, they were quite similar in terms of the gross flows of energy. And probably even there's every reason to believe that the primary productivity figure he used for the North Sea was too low. So in terms of gross energy flow, the systems weren't that different. In terms of species, they're extremely different. And then I'll give you a couple of examples, if you can hold out, of where we've used macrobenthos in environmental studies that can be used maybe more management relevant, perhaps. One is oil pollution research that I participated in at the Marine Ecosystems Research Laboratory at the University of Rhode Island, which I did as a postdoc. They looked at oil pollution in large mesocosms. These are tanks which, as you see, are about 6 meters tall, 5.5 I think actually, and about 2 meters in diameter. So they're on the order of 14 cubic meters of water or so. And at the bottom we have a sediment tray which is about 2 meters in diameter, 30-40 centimeters deep sediment, so they weigh a few tons, and which we filled with fauna in Narragansett Bay and then transported to the lab and put in the bottom of the tanks, and then we filled up with water from Narragansett Bay. And then they would go for a while. There were 14 of them, and then we started when we saw that there was biological activity going on in a reasonable way, then we started adding oil to some of them. Small quantities of oil were suspended in water, in total about 100 parts per billion, 0.1 part per million, which were added over some weeks. And in individual studies, it could be shown that this was negative for phytoplankton. It was negative for zooplankton and for benthos. No one liked oil when you added it. But because the oil reduced the numbers of zooplankton and particularly filter feeding benthos, we saw in fact an explosion of phytoplankton. Even though the phytoplankton tested by themselves weren't particularly happy about the oil, they still proliferated and became the water became quite green. And we also saw down at the bottom, we saw benthic diatoms developing a film over the bottom because there was nowhere, they weren't being eaten anymore. And the most sensitive ones were the amphipods and the ostracods. I worked with some other people who looked at the macrofauna. I looked at the meiofauna. Then we stopped adding the oil, and we saw that the first ones to recover were the harpacticoids. So this was an early example of trophic cascading. While I was in the US doing this work, there was an oil spill in the Baltic, the Tsesis oil spill. The Swedish authorities had mapped the inlet to Södertälje, and they had actually found a rock sticking up, but they had forgotten to put it into the sea charts. And this was found by a Soviet tanker. And there was a spill of something like 700 tons of oil or so. And which covered, here's Asker, and it covered quite a large area. And the people at Asker, they had a big research project on the ecosystem of the Baltic, but they dropped everything and started collecting samples here. And we actually had some cooperation with the NOAA team that came in and did the analysis of the oil, the chemical analysis of the oil. Something like 1,000 tons were leaked, but a fair bit was cleaned up. Maybe 300 to 500 tons remained. And they covered things like bacteria, phytoplankton, zooplankton, fish, and so on. And in fact, what they found was that oil reached the benthos quite rapidly. Within a day, within a couple of days, you had oil on the bottom. And before, the general idea was that oil is lighter than water. It would remain in the surface, it would reach the coast, and maybe once it had reached the coast, it would incorporate sand and so on and start working out from the coast. It turned out that because it's finely divided, it will stick to very small particles that are in the water and they sink. So in fact, it reaches the bottom quite rapidly. And there, meo and macrofauna started dying, particularly the amphipods and the ostracods. And then we followed this over time, and we saw that harpacticoids recovered first, just like in the mesocosms. And when the amphipods were gone, macomas could recruit in the next spring. And a little over a year after that, they were big enough to be found in the macrobenthos in quite large numbers. So we could see an entire change. And of course, macomas live for up to 27 years. So that one instance would probably have an effect that would endure for at least 10 years or so. But it turned out that we followed that quite carefully for some while. And then we had a year, I think, in 84, I think it was, after 6-7 years, we had a year when this area was affected by oxygen deficiency. And then we couldn't tell whether it was oil or eutrophication that decimated the fauna. So then we stopped following up. It was meaningless. And in the plankton, we could see that pelagic bacteria and phytoplankton seemed to increase soon after the spill, whereas the zooplankton became fouled with small oil particles, and apparently either died or at least stopped feeding. So you had the same kind of we had the same kind of trophic interactions as in the mesopocosms. What we also saw was that phytobenthic snails and crustacea were greatly reduced after the spill. That's probably where the highest concentrations of oil was, because you had an oil, surface oil, floating up to the coast. And next spring we saw fewer spawning herring in the spill area, and those that spawned had lower hatching success than in other years. And as you say, in follow-up ended in 1984. By that time we could see considerable recovery of the phytobenthos, and we could see that the macrofauna had started to come back, but we still had many more macomas. And then, of course, oxygen deficiency ended that. So here you have an instance where you can use the macrobenthos to say something about the impact on the ecosystem, and you can also see something about the how long the impact endures, because these are long-lived organisms. Oxygen depletion in the Baltic is a common problem. This is approximately the way it's been for a long time. You have in the Baltic a number of areas where there is no oxygen at all, and others around that an area where the oxygen exists, but in such low quantities that sensitive organisms are not able to live there, and things like fish avoid the area. And this is something that's developed over time. From here you're at 1890, then you had oxygen everywhere. 1932, the first instance where they actually found zero oxygen. 1950s was the first time they actually took sediment macrofauna samples and found nothing in it in the Baltic proper. And then 1951 we had a big inflow of saline water, which just sat there for a long time, and you started to get into this downward spiral when it combined with eutrophication. And in the 1980s, we returned to a number of stations that had been sampled in the 1920s by one of the first people who worked in the Baltic with quantitative sampling methods. This was two researchers, Hessle and Wallin, who took samples in the 20s. And Hessle in the northern part of the Baltic and the Bothnian Gulf of and the Bothnian Gulf of Bothnia, Valin in the southern part of the Baltic off Scania and so on. And we returned in the late 70s to the same stations as well as we could. What we did, we followed their descriptions. There was, of course, no GPS. But we followed their descriptions of where they were, and we looked with the echo sounder for the right depth, and then we checked that we had a sediment that was of the type described by them. When those three things happened, we considered that a replicate of their sample. And then we looked at, we took a ratio, the biomass in the 70s divided by the biomass in the 20s. So if the figure is 1, there has been no change. If the figure is above 1, there has been an increase in biomass. If it's below 1, there has been a decrease in biomass. And then we plotted this versus depth. And this is the picture that emerged. Of course, we expected that below the halocline, we have, on the whole, much lower biomass than they had in the 20s because of oxygen deficiency that has wiped out most of the macrofauna. But what really surprised us was that on most stations there was now much more biomass. And this is a logarithmic scale. So you're talking here about 5 to 10 times more. So it's a very dramatic increase. And we actually used the same type of Peters and Grab. We managed to find one that they had used. So it's not a method thing. And the only explanation for this we could come up with, and I think it still stands, even though there has been some debate, is that because of eutrophication, there's much more food produced in the Baltic today. And this was the first demonstration of a biological response to the nutrient enrichment we knew at that time was going on in the Baltic. We had seen, of course, oxygen deficiency, but this was the first change in the biology that was reported. Later on, of course, the Kautsky family returned to diving transects and found similar changes in the phytobenthos. So there are changes, and a few years later also Anders Stigebrand did a very clever thing in that he looked at oxygen in the water column that had been measured on Swedish lightships. We had, in a number of places in the Baltic in those days, we had lightships to guide ships. And the people who worked there had very little to do. They were just sitting there and seeing that the light was on. They had nothing else to do. So things were found for them to do. They were asked to measure oxygen concentrations in the water repeatedly over time. So they would take samples of oxygen, and it was used for absolutely nothing until 30 years or so later, when Anders Stigebrand came up with a brilliant idea that oxygen is produced by photosynthesis. So if there's a lot of photosynthesis, we should have more oxygen in the water. And he could calculate the increase in primary productivity from that, and again show that yes, indeed, we have a very clear response to eutrophication. I'll just skip these. I think that this is about as far as we get today. Okay, thank you. And any questions?